Cell cycle regulation in a cell. Chrissy Wilhelm. In this presentation, we're going to define the stages of the cell cycle, explain how cyclins and cyclin-dependent kinases regulate transitions, identify major checkpoints and their roles, discuss the role of tumor suppressors and oncogenes, and relate disruptions in regulations to cancer. Cell cycle regulation is essential for maintaining the accuracy and stability of cell division. During the cell cycle, checkpoints carefully monitor processes such as DNA replication, repair, and chromosome separation to ensure that errors are corrected before the cell progresses to the next phase. This precise control guarantees that each daughter cell receives an exact copy of the genetic material, preventing mutations that could compromise cellular function. By ensuring faithful DNA replication, cell cycle regulation plays a critical role in growth, development, and renewal of tissues. Beyond genetic accuracy, proper regulation is also crucial for overall tissue health. When cell division occurs in a controlled manner, tissues maintain the right balance of growth and repair. Conversely, improper regulation can lead to uncontrolled cell proliferation, as seen in cancer, or failed cell division, which can contribute to developmental disorders. Cell cycle regulation not only preserves genomic integrity, but also protects the body from disease. The cell cycle is a highly organized series of events that ensures cells grow, replicate their DNA, and divide correctly. It is divided into several distinct phases: G1, S, G2, M, and G0. Each phase plays a critical role in preparing the cell for division and maintaining healthy tissue growth. In the G1 phase, the cell grows in size and produces RNA, and synthesizes proteins necessary for DNA replication. This is a major checkpoint where the cell decides whether to continue through the cycle or enter a resting state. Next, during the S phase, DNA is replicated, ensuring that each daughter cell will inherit an identical set of chromosomes. The G2 phase is the second growth period in which the cell prepares for mitosis by producing proteins required for chromosome separation and finalizing DNA repair. The M phase, also known as mitosis, involves the actual division of the nucleus and cytoplasm, creating two genetically identical daughter cells. Some cells enter G0, which is a resting state, where they no longer divide, but carry out their specialized function, for example, a nerve cell or a muscle cell. This diagram represents the phases in a circular flow, emphasizing the continuous and cyclic nature of cell growth and division. The diagram highlights the checkpoints at G1 or S, between G2 and M, and during mitosis, which ensure accuracy and prevent damaged DNA from being passed on. This underscores the cell cycle's importance in growth, repair, and maintaining genetic stability. Regulatory proteins play a critical role in controlling the cell cycle, ensuring that each phase occurs in the correct order and at the proper time. Cyclins act as regulatory subunits that bind to an active cyclin-dependent kinases, which are enzymes responsible for phosphorylating target proteins involved in cell cycle progressions. CDKs on their own are inactive. They require the presence of specific cyclins to function properly. Importantly, cyclin levels within the cell rise and fall in an oscillating pattern, and this fluctuation directly regulates the timing of CDK activation. For example, certain cyclins accumulate at a specific point in the cycle, triggering transitions such as from G1 to S phase, or from G2 to M phase. Once their role is complete, the cyclins are degraded, leading to inactivation of the CDKs until the next cycle. This tightly regulated system ensures orderly cell division and helps prevent errors such as uncontrolled growth or incomplete DNA replication. Checkpoints are essential control systems in the cell cycle that act as quality assurance mechanisms to preserve genomic integrity. The first major checkpoint, the G1S checkpoint, ensures that DNA is intact and suitable for replication, preventing the cell from advancing to the S phase if damage is present. Later, the G2 to M phase checkpoint verifies that DNA replication has been fully completed without errors, allowing the cells to safely enter mitosis. These early checkpoints act as safeguards to prevent damaged or incomplete genetic material from being passed on. The final checkpoint, known as the spindle checkpoint, functions during mitosis by confirming that all chromosomes are properly attached to the spindle apparatus and aligned at the cell's equator. This guarantees that each daughter cell will receive the correct number of chromosomes. If any checkpoint detects problems, the cell can pause the cycle to make repairs, or if the damage cannot be corrected, undergo apoptosis, or programmed cell death. Together, these checkpoints act as decision points that maintain genomic stability and prevent the uncontrolled growth of a characteristic of cancer. Tumor suppressors are proteins that act as safeguards against uncontrolled cell growth by regulating the cell cycle and preventing the accumulation of genetic damage. One of the most important tumor suppressors is p53, often referred to as the guardian of the genome. P53 plays a central role in detecting DNA damage and responding appropriately by pausing the cell cycle, activating DNA repair mechanisms, or triggering apoptosis if the damage is beyond repair. By doing so, P53 helps maintain genomic stability and protects the cell from passing mutations to future generations. Another key suppressor is the retinoblastoma, or RB protein, which regulates the critical transfer from the G1 to the S phase of the cell cycle. RB functions by controlling the activity of transcription factors needed for DNA replication, effectively acting as a checkpoint to ensure the cell only progresses when the conditions are appropriate. Mutations in either p53 or RB can have serious consequences as they disrupt the balance of the cell cycle regulation. Loss of these tumor suppressor functions allows cells with damaged DNA to continue dividing unchecked, greatly increasing the risk of tumor development and cancer progression. Oncogenes are mutated forms of proto-oncogenes, which normally function to promote healthy cell growth and division in a controlled manner. When these genes become mutated or abnormally activated, they can drive excess and uncontrolled cell proliferation, contributing to the development of cancer. Two well-known examples are the RAS and the MYC. Mutations in RAS can cause the protein to remain permanently active, sending continuous signals for cell growth, while overexpression of the MYC leads to unregulated transcription of genes that stimulate cell cycle progression. These changes disrupt the normal balance of cell regulation, pushing cells to divide even in the absence of proper signals. Oncogenes contrast with tumor suppressors, which act like brakes to slow down or stop the cell cycle when errors or damage occurs. When oncogenes accelerate cell division, tumor suppressors like p53 and RB function to prevent abnormal growth. Cancer often develops when both systems are compromised. Oncogenes are turned on permanently, driving proliferation, while tumor suppressors are turned off, removing critical safety checks. This imbalance between the growth promotion and regulation is a hallmark of cancer biology. Signal transduction is the process by which cells respond to external signals such as growth factors by activating specific receptors on their surface. When growth factors bind to these receptors, typically the receptor of tyrosine kinase, it triggers a cascade of intracellular signaling pathways. Two major pathways involved are the MAPK and the PI3K pathways. These cascades transmit signals from the membrane to the nucleus, ultimately affecting the activity of proteins that control cell cycle progression, particularly cyclins and cyclin-dependent kinases, which are critical for driving the cell through the various phases of the cell cycle. These signaling pathways integrate external environment cues with the internal machinery of the cell cycle. Depending on the strength, duration, and type of signal received, the cell may either proceed with division or pause the cycle to repair damage or await better conditions. For example, if the MAPK path is strongly activated, it can lead to increased production of cyclins that push the cell from the G1 phase to the S phase. Conversely, if the signals are weak and stress is detected, inhibitory proteins might be expressed to halt the CDK activity and prevent cell cycle progression. This integration ensures that the cell only divides when conditions are favorable, maintaining proper tissue growth and preventing uncontrolled proliferation, such as in cancer. Apoptosis is a highly regulated process of programmed cell death that allows cells to die in a controlled and non-inflammatory manner. Unlike necrosis, which is a form of traumatic cell death caused by injury, apoptosis involves a series of biochemical events leading to characteristic changes such as cell shrinkage, chromatin condensation, DNA fragmentation, and membrane blooding. These changes are orchestrated by a family of enzymes called capsases, which are activated in a cascade and work to dismantle the cell from within. The cell is then engulfed and removed by immune cells, such as macrophages, without causing harm to the surrounding tissue. Apoptosis plays a critical role in maintaining proper control over the cell cycle by eliminating cells that are damaged, no longer needed, or potentially dangerous. For instance, if a cell has DNA damage that cannot be repaired, tumor suppressor proteins like p53 can trigger apoptosis to prevent the cell from progressing through the cycle and dividing uncontrollably. This safeguard helps prevent the accumulation of mutations and reduces the risk of cancer development. Additionally, cells that fail to pass the critical checkpoints during the cell cycle may be directed to undergo apoptosis, ensuring that only healthy and properly functioning cells are allowed to proliferate. The balance between cell proliferation and cell death through apoptosis is essential for tissue homeostasis. During development, apoptosis helps shape organs and removes unnecessary structures. In adults, it maintains the correct number of cells in the tissue and allows for turnover and regeneration without disrupting the tissue architecture. If apoptosis is too active, it can lead to degenerative diseases, while insufficient apoptosis can result in uncontrolled cell growth, as seen in many cancers. Thus, tight regulation of both cell cycle and apoptosis is vital for the health and stability of multicellular organisms. Dysregulation of the cell cycle is a key factor in the development of cancer. Normally, the cell cycle is tightly controlled by checkpoints, regulatory stages that ensure these cells only divide when they are healthy and conditions are favorable. These checkpoints monitor DNA integrity, proper chromosome alignment, and sufficient cell size. If problems are detected, the cell cycle is paused to allow for repair, and if the damage is too severe, then it triggers apoptosis. However, in cancer cells, these checkpoints often become non-functional due to mutations in key regulatory genes. The loss of checkpoint control allows the cells with DNA damage or mutations to continue dividing, leading to genomic instability and tumor formation. Oncogenic mutations, change in genes that promote cell division or survival, play a central role in this process. These mutations often act as proto-oncogenes or tumor suppressor genes. When the proto-oncogenes are mutated, they can become permanently activated, leading to constant cell division, even in the absence of the growth signals. At the same time, mutations that inactivate tumor suppressor genes, such as the p53 or the RB, remove crucial barriers to unchecked growth. This imbalance between cell cycle promotion and regulation, again, is the hallmark of cancer. A well-known example of this kind of dysregulation occurs in infections with high-risk types of human papillomavirus, HPV, such as HPV-16 and HPV-18. These viruses produce viral proteins E6 and E7 that specifically target and disable two major tumor suppressors. The E6 binds to promote the degradation of p53, while E7 binds to inactivate the RB. By disabling these proteins, HPV-infected cells lose the ability to properly regulate the cell cycle and undergo apoptosis, allowing them to divide uncontrollably. This mechanism is a major driver of cervical and other HPV-associated cancers, illustrating how viral oncogenes can hijack cell control and contribute to cancer development. Understanding the role of cell cycle regulation in cancer has led to the development of targeted therapies aimed at correcting or blocking dysregulated pathways. One major class of these therapies includes CDK inhibitors, which block the activity of the CDKs, key proteins that drive the cell cycle progression. For example, CDK4/6 inhibitor is used to treat certain kinds of hormone receptor positive breast cancer. By inhibiting the CDK4/6, this prevents cancer from progressing from the G1 to the S phase of the cell cycle, effectively slowing or halting tumor growth. This approach can be especially effective when used in combination with hormone therapies that reduce tumor-promoting signals. Another important therapeutic strategy involves targeting growth factor signaling pathways that are overactive in cancer cells. Many tumors rely on continuous signaling through pathways like the MAPK or the PI3K, which promote survival, growth, and division. Drugs that block these receptors, or downstream signaling components can interrupt these cancer-driving signals. These treatments aim to cut off external and internal cues that cancer cells misuse to sustain their rapid proliferation. Looking forward, the future of cancer therapy lies in precision medicine, tailoring treatments based on specific genetic and molecular features of an individual's tumor. Advances in genomic sequencing allows clinicians to identify key mutations or pathway alterations in the patient's cancer and match them with drugs that specifically target those changes. This personalized approach increases the effectiveness of treatment while minimizing side effects. Ongoing research is also exploring new targets in cell cycle regulation and resistant mechanisms. offering hope for more durable and precise cancer therapies in the years to come. So to summarize, the cell cycle is a highly regulated process that ensures cells grow, replicate their DNA, and divide correctly. This regulation is maintained through the coordinated action of cyclins and CDKs, which act as molecular switches to move the cell through different phases of the cycle. Checkpoints, such as G1S and G2M transitions, act as quality control mechanisms, ensuring that DNA is intact and fully replicated before the cell progresses. These safeguards prevent the accumulation of mutations and maintain genetic stability. Tumor suppressors and tumor suppressor genes and oncogenes play key roles in controlling this balance. Tumor suppressors such as p53 and Rb function as brakes on the cell division, halting the cycle in response to damage or stress. Conversely, the proto-oncogenes normally promote health and survival, but when mutated into oncogenes such as the RAS or the MYC, they can drive uncontrolled proliferation. Together, these systems work like a balance between acceleration and braking in the cell cycle. When the regulation breaks down, the consequences can be severe. Mutations, mutations that inactivate tumor suppressors or activate oncogenes lead to unchecked growth, allowing cells to bypass checkpoints and accumulate genetic errors. This dysregulation is a fundamental hallmark of cancer, enabling cells to proliferate uncontrollably, resist apoptosis, and eventually form tumors. Understanding how these molecular regulators interact provides important insight into both normal cell function and the development of disease. If you'd like to check out any of the resources for further information, please check out these four.